Welcome to this morning's presentation on the applications of mass spectrometry-based proteomics in research. We've got three main sections in the program. Introduction to mass spec-based proteomics, and that would be presented by Pierre. Typical experimental applications of mass spec, presented by Rowan, and bioinformatic analysis and visualization of proteomics data, presented by Harinder. So now I'm going to hand over to Pierre, is our platform services manager and our expert in proteomics, sample preparation and mass spec analysis, and he'll introduce that to you. Thanks, Pierre.
So yes, thanks for the introduction, Christopher. So I'm going to talk about mass spectrometry-based proteomics. And okay, I have to do it like that. So what I was going to start with is just to give you a definition of proteomics. So proteomics is the analysis of the entire protein complement of a cell, tissue, or organism under a specific defined set of conditions. So examples of proteomics analysis could be, if you will look at protein interactions, like for example here, these two protein membranes that would interact with each other. So that could be an example of proteomic analysis. Protein that will be at the membrane that could be translocated inside the cells or in the mitochondria. One could also look at signal transduction of protein that could be, for example, like here, phosphorylated, but that could be many other post-translational modification. I'm thinking acetylation, glycosylation, and there's many others. So that could also be one example of proteomic analysis. What we can also look at is protein turnover. So protein could be degraded with time or by the addition of drugs. And these are examples of proteomic analysis that can be performed. What I'm showing here is an example. So this is a proteomic analysis that could be done on the tumor necrosis factor signaling. And this is just to show you how complex a proteomic analysis can become. Because here, things are linked together and protein changes will happen at three different levels. We have protein-protein interactions, that could be from the outside of the cells to the membrane, but also formation of complexes inside the cells, or even in the nucleus. We are also a lot of regulations done at the PTM levels with phosphorylations at many different sites or many different proteins. And we also have protein quantities that are changing all the time. So we have the apoptosis that is happening in this signaling pathway, but we also degradation of a lot of proteins happening. And the protein quantities are also changing because you have proteins that are imported from the outside of the cells. Some proteins that are in the cytosol are now translocated inside the nucleus, and all these degradations happening. So this is an example of proteomics analysis and how it can become easily very complex. So usually researchers are more used to genomic analysis, but there are many examples where proteomics become a necessity and where genomics is not going to help you. So as I mentioned before, if you were looking at protein-protein interactions, genomic is not going to be of any help to you. Again, as I mentioned before, if you will look at detection of post-translational modification on site of proteins, genomic is also not going to help you. And more, the researchers are interested into the detection of biomarkers in body fluids. That could be serum, that could be urine. And only in this case, genomics is not going to help you, but proteomics is the only approach possible. Moreover, there is more and more evidences that the level of RNA changes does not translate directly into the similar protein changes at the protein level. So it's really important in this case to have a look at the changes at the protein level, because mRNA is probably not going to give you a very accurate measurement. So historically, proteomic studies were done by 2D page electrophoresis. We're talking around 20 years ago. And I remember when I was doing my PhD, I did many, if not hundreds of those 2D pages. So let's imagine that you will have to study E. coli, and you want to study the effect of heat shock. So you'll have two sets of cells prepared on the normal conditions and on the heat shock. So you will harvest your cells, you will prepare your proteins. And what you will do in this case is that you will do two separation. The first separation will be protein separated according to the isoelectric focusing point, so it's first dimension. Then you will take this strip, because it's done in a strip, and you put it on top of a gel, and you will then run the second dimension, will be SDH page. So the gel will be then stained, commercially stained or others, and you will have to compare your two conditions. So you have two sets of gel, and you try to compare what's happening from one gel to the other. These spots are getting stronger or disappearing. But those approaches have a lot of limitations. First will be the low resolution. As you can see in this gel, a lot of spots are on top of each other. So it's very hard to distinguish distinct spots. The reproducibility was also a problem, because it was so many steps involved that it was very difficult from one experiment to the other to be highly reproducible. And that was, of course, a problem to be able to conclude from your experiment. The narrow dynamic range, because here maximum a couple of hundred spots, and if you would know that the proteome of humans will involve more than 20,000 proteins, we are so far from seeing these thousands of spots. So again, narrow dynamic range was really a problem. But mainly as well, the problem with those 2D pages were the fact that hydrophobic proteins were extremely difficult to be analyzed. They had a tendency to aggregate in the first dimension, and you will never transfer them into your second dimension. This is why researchers sort of moved away from these 2D pages and tried to develop new methods. And I'm going to introduce you to mass spectrometry-based proteomics that evolved from these 2D pages. So what I have here is a representation or a graph that shows the number of proteins that were identified with the time. So here we're talking 10 to 20 years ago. And what we have here in blue are the sample preparation methods. We have in green, we have the hardware, and in this pinkish red, it's more about the software. So 10 to 20 years ago, we were able to identify around, let's say, 200 to 500 maximum proteins. So nowadays, now if you will look at what we are able to identify today with a good sample preparation, we are more into the 5,000 protein identified per runs. And this is not really due to the evolution in sample preparation, because we are still using shotgun proteomics. We are still using SciLac or MRM. This is also not due to the fact that the evolution in the software has been extremely useful, because Mascot here and even MaxQuant are still the gold standard. It is really due to the fact that the hardware have been becoming faster, more sensitive. And what I wanted to say here is that La Trobe University has been very lucky that this year we have been successful in a LEAF grant, and we have installed the latest instrumentation from Thermo Fisher with the orbitraffic clicks. So we are now able to identify thousands of proteins per run. Another reason why I wanted to introduce you to this graph is I wanted to talk about shotgun proteomics. So shotgun proteomics was introduced more than 20 years ago, and it's still used in probably 99% of the facilities that are running proteomics. And in the year 2001, when this technique of sample preparation was introduced, there was really a jump into the number of protein identified. So we are around the 100 marks, and we jumped to the 300 to 500 number of protein identified. So what is shotgun proteomics? And again, this is really a technology that every lab is using. So it's really important that you at least have the notion of understanding what shotgun proteomics is about. So shotgun proteomics is also called bottom-up approach. So this is a technology or a sample preparation method where we'll start with cells or tissue and we'll extract the protein. So that could be done with detergent or caltrops like urea or other reagents. So we extract the proteins, and the proteins need to be digested into peptides. So what sort of enzyme are we using here? I will say in most cases we are using trypsin. Why trypsin? Because trypsin is very efficient and very specific. And also, so you might know that trypsin cuts after lysine and arginine. And the number of lysine and arginine in proteins are appearing so that usually the mass of the peptides or the mass of the peptides that we obtain are between 6 and 15 to 25 amino acids, which is exactly what the instrumentation needs. So it's not only specific, it's not only very efficient, but it's also very useful because it gives us the right amino acid sequence in terms of the number of amino acids. So what are we doing after we obtain our peptides? Is that the peptides are not directly injected into the instrumentation, but it needs to be separated first into a liquid chromatography system. With the time, usually the type of column that we are using is rely on hydrophobicity of the peptides, and we are most of the time using C18 type of columns. So the peptides are going through the columns, they elute with the time according to the hydrophobicity by applying a gradient of usually acetonitrine. So they, before they're introduced into the instrumentation, because we are in the liquid phase from the HPLC, we are doing an ionization step where the peptides that were in liquid form are now in form of a gas phase, and this is what is introduced into the instrumentation. So what the instrument is doing two things. Is taking the mass of the peptides, this is the first thing, and then the peptide is fragmented. So it's important to understand that. Two experiments done in the instrument, the mass of the peptide is followed by the fragmentation of the peptide. And for a typical sample nowadays, we are talking of hundreds of thousands of those little experiments. So you can imagine the complexity. And so an experiment will happen maybe in an hour. And at the end of the day, at the end of this hour, we'll obtain hundreds of thousands of little fragmentations. So of course, the next step will be to make sense of all of that. And this is done by informatics, basically. So imagine that we have our fragmentation. The fragmentations need to be put into peptides, and the peptides now need to get back into proteins. So you can imagine this big puzzle, this jigsaw puzzle made out of hundreds of thousands, and if we even consider a full proteomics analysis, we are talking of millions of these little pieces of jigsaw puzzles. So really the bioinformatics become a very important part of the analysis. So, at the end of this experiment here, what we have is the, not the quantities, but we have just identified the proteins. What is important to understand is that mass spectrometry by itself is not a quantitative technology. And this is quite interesting to understand. Sorry, it's quite important to understand that. So imagine that you will take one protein, and this protein, so you will unfold it before you produce the peptides. So now that I have obtained all my peptides after the addition of trypsin, and I'm going to analyze those peptides, even in our latest technology, if you will take those peptides coming from this single proteins, you will see that all these peptides, even though they have different masses, they have different intensities. So understand that you cannot use the intensity of the peak coming from the peptides to be a good indicator of the amount of protein in the sample. You understand all those peptides, they come from the same proteins, they all have different intensities. But what we can do in order to get quantitative information is to look at the differences in peak intensity of the same peptide between multiple samples. Yeah, so you will look at the intensity of the same peptide across different samples. And by looking at the different intensities between this same peptide, you can then have information about the relative quantity between these different samples. And this is why, and this is how different workflows have been developed in mass spectrometry, and they all rely on comparing samples with each other. We don't have absolute information of quantities. We have relative informations. So in this graph, we have in blue and green, we have two different experimental conditions, so two different samples. And this, this analysis, or this, I call it, this comparison, it can be made at different levels, either at the tissue or cell levels, where you might have heard about SALAC, which is when you will mix your sample very early on by growing your cells with heavy isotopes. So you will mix your sample very early on. The comparison, again, can be done a bit later on at the protein or at the peptide level by chemical labeling, where you will mix your sample after you've chemically treated your different samples, and then you do the comparison at that level. Or it can be done very at the very latest step at the data analysis level if you will use a workflow called label-free. So it's really, there is no mixing, and the comparison, the relative quantities are, information is produced only at the data analysis level. So that will be the end of my little presentation here, and I was just, yeah, not really details, but just to give you more or less rough ideas about what mass spectrometry can be applied to give you quantitative information and how you will conduct your proteomics analysis.
So I'm going to cover a few common scenarios for beginners to the platform. And I guess one thing I just realized listening to Pierre's talk was we're about much more than just collecting a sort of typical proteome where you try and collect all of the proteins from a sample. We also do quite targeted stuff, which is actually our more common, some of our more common work, especially during the semesters when students are busy in the lab. So 4 common scenarios I'm going to cover. One would be, I have purified a recombinant protein in the lab, so I want to check its identity before I do lots of experiments with it. Second one would be, I've precipitated a protein complex, which indeed came up in the question time just before. Third scenario would be, maybe you made a knockout mutant in the lab and you want to see what else has changed in that organism as a result of your mutation. Or 4, the final one that I'll cover would be, I'm interested in post-translational modifications, which also came up in question time. So it's nice that we're at least hitting some of the topics that people are interested in. And it's just going to be very entry level. As Pierre mentioned, you come in, we'd love to sit down and talk to you about the really fine details of planning your experiment. But this should get you inspired and sort of in a position where you sort of know what you can and can't do. So moving on, in that first scenario, researcher has purified a recombinant protein. We have 2 useful techniques here that will help you out in that case. One is our intact mass analysis, and our second technique is peptide mapping. So when you start thinking about these techniques and working with us, there's a few considerations that you sort of just have to have in your mind. And that is what they can and can't do. So for intact mass analysis, what this will do is if you have a pure protein, it will give you a very accurate mass reading for it, surprisingly enough. So in the Daltons, in this little example on the right here, this protein is 8,560 Daltons. So it's much more accurate than an SDS page gel can provide you. Some restrictions, some proteins may not be compatible with the technique, so it's not always guaranteed to work for everything. And again, you'd have to have that pure protein. If it's a mixture, intact mass analysis is not going to be so great because they may interfere with each other. Second technique covers a lot more situations. We can take an in-solution sample or even a band cut from an SDS page gel. You can analyze that. And what this tells you is what peptides are present in that protein band, which is very useful to verify you've got the right clone, the right plasmid, whatever you started with is indeed what you ended up with. Some considerations would be, does your protein actually digest nicely with trypsin? If you have a very unusual protein, say it's extremely small, repetitive, we may not get as many peptides as usual, but generally it works very well for the vast majority of stuff people submit to us. And in peptide mapping, in this little diagram, you've got a red bar here representing the sequence of your protein, and these little gray peptides map to different regions of the protein and would tell you that, oh yes, in this case, I have actually identified most of my protein is there, it's present, it's got the right sequence, I'm good to go with further experiments. So some of the questions people might come up with in this case is, oh, I think my protein is a bit small, it's running fast on my gel, I'm a little bit suspicious of it, or maybe I didn't purify as much as I expected to. So in that case, you know, things aren't aligning quite right, you want a bit more confirmation. So yeah, you can supply, for peptide mapping, you can supply your proteins in solution. If it is not pure and you've got a mixture, it's probably best to separate it by SDS page and you'd cut a single band out. Keep in mind though, our instruments are very sensitive, and even when you think you've cut out a single band, we have, you know, can find 100 or more proteins in single bands, and that's basically just because SDS page doesn't separate things out as well as it looks by Kamasi staining, and there's traces in there that we can still pick up. But generally, as always with mass spec, the purer the protein sample is, the better for us. And then that second one, intact mass analysis, as we said, this is just gives you a very accurate and precise number quite quickly. And the really great thing about that mass analysis is that you can tell if even one amino acid has changed according versus your expected sequence. So very accurate, but it won't tell you what has changed. So it's really just a quick check. It'll give you a number, and then you figure out what to do after that point if it doesn't align with your predictions. So here's a little workflow for if you have, you would have heard me mention whether you have a protein as in a gel band or in solution. So that really restricts what techniques we can use. So if you have pure protein in solution, you have two options. You could take this top path to get an intact mass, nice accurate mass for your protein to confirm its identity. Or you can also go through the peptide mapping route and confirm your protein's identity that way by mapping peptides to the predicted sequence. Whereas if you just have a gel band and maybe, yeah, you've run it out, it's not particularly pure, so you use SCS Page. In that case, you can't do the intact mass. So if you've got a gel band, we'll just run peptide mapping for you to confirm the protein identity. So the second scenario I mentioned was a researcher who has affinity purified a protein complex, say a pull down assay, an IP, something like that. And the question here is, what is my protein binding to? What is in that complex? Is anything in that complex? We can supply a simple protein analysis for these types of experiments, which will give you a report of the proteins that are present in that sample, which would be in your complex. Important considerations if you are going to proceed with this sort of experiment would be, do you have enough protein to detect? Sometimes when you do a pull down, the interaction is quite weak and it may not have a lot of the bound complex present. So generally in these situations, you want to have something that's visible by a Comasi stain gel, just to make sure there is enough there to catch everything in the complex. Some things you can do beforehand is have a look at the literature to see try and get a handle on how abundant your complex, predicted complex partners are in other studies. You can have a look on UniProt. Some model organisms have quite a lot of information on protein abundance available. UniProt's an online database. It's pretty easy to search for your protein name or blast to it. And one other consideration for the complex is you can submit your protein on beads if you want to. So that first point was, do you have enough protein? And this comes down to the same sort of things again and again, is do you have enough to detect in the mass spec? And so how abundant are your binding partners? You might have enough of your initial complex partner, but the partners may be extremely rare and you might need to use quite a lot of input lysate to capture enough to detect. So you need to think about this whole abundance story. And one thing I will also mention now that I thought of during question time on the protein complex is that the platform also houses 4 instruments that are not mass spec based, which can analyze protein-protein interactions. So you can come and talk to us and we'll tell you all about those instruments for protein-protein interactions, and some of those can help you characterize a complex. Third scenario, I made a mutant, so knocked a gene out of some particular organism and you're interested in whether it has regulated other protein abundances in the cell, have things gone up or down in response to this perturbation. So in this case, we can run a differential abundance analysis of a proteome. So you'd get a report of all the proteins that are detected in your samples and where possible, a relative change. So if it was detected in all of your treatments, you'll get a nice fold change, quantitative measurement for those. And these are relative changes though to your controls. And you can get some statistics on that. So that's really handy. In these cases, what you need to consider for your experiment is adequate replication and controls. So replication is always really critical if you want good statistics, and controls are also really important so that you can make good inferences from your data. Another key consideration here is that, again, we have to be able to detect your proteins of interest before you get too far ahead. If you are really interested in the change in some particular kinase or something, if we can't find it with our standard method, then we won't be able to quantify it across all of the samples. So as usual, we're very sensitive instruments, but as Pierre said, we're still finding at best around 5,000 out of 20,000 proteins in the human proteome. So you want to make sure your protein of interest is in that zone before you get too far ahead. Fourth example is someone who would be interested in post-translational modifications. We can definitely do that work, and in this case you might be interested, the most common post-translational modification studied is probably phosphoproteomics, where you're looking at phosphorylation of proteins by kinases, for example. There are other examples such as glycosylation, lipidation, or even just cleavage of a protein by proteolysis to activate it. So in this case, the considerations are how do you enrich for your PTM of choice? This is because, again, you're up against the abundance and sensitivity problem. So most proteins have low abundance and are hard to detect. And then on top of that, you're adding a post-translational modification, probably not to all of that protein population. So they become even rarer. It's the rarest of the rare in some cases. In this little diagram on the right here, I've just to illustrate the concept for you, is in a complex solution of proteins, you know, only some have the modification of interest. So you need to be able to pull those out and perform what we'd call enrichment. So the question becomes, how can you enrich your target enough to detect it? And we certainly have techniques to purify, for example, phosphorylated proteins. But again, it all comes back to basically, do you know what modification you are looking for? Do you know how the protein is modified? That definitely helps us plan an experiment. And this is definitely something where you would 100% come in and have a good sit-down meeting with us to plan the strategy before you get really stuck into your experiment. Right, I think to summarize those 4 techniques, we'll go through this again. So I mentioned intact mass analysis. This is really handy for a quick and accurate measurement of the mass of a pure protein in solution. And again, there's a few caveats on that, but generally you want a pure protein and in solution, so you can't do it with a gel band. For peptide mapping in simple protein ID, the second technique, here you'll confirm the identity of a protein in solution or from a gel band. So this would get you that real confidence that you've got the right thing and you can move ahead with your other experiments. And in this case, you just want to make sure that your sample is pure enough that we'll get lots of signal for it. The third example, differential abundance. This is great for quantifying relative amounts of proteins in your sample, usually to compare different treatments. First, make sure that you can actually detect your protein before you start worrying about how the relative amounts of it there are. And then finally, more specialist techniques for PTMs would be, an example is phosphoproteomics. And in this case, enrichment of the PTM is often critical for success. So you need to know how much of it roughly you've got, and that will help tell us how much we need to enrich for detection. So one other question that came up in question time was how to contact us and how do you get started. So this final slide is just a little summary of how the general process is. And normally people will just email us or they could phone or drop into the lab in person. But generally an email is enough just to get in touch and say, hey, I'm interested in doing some proteomics. This is what I'm up to. And generally we'll set up a meeting with all of us or whoever is most relevant expertise. And you'll get some good solid advice that's specific to your experimental problem. And this really means that you'll start off your experiment on the right foot. You can then go away, perform your experiment, and once that's finished, you would complete a sample form, which I've got a little picture of down here for us, send that in, and that helps us make sure we've got the exact details for that, what actually happened with that experiment. And it's really important for us in terms of making sure we choose the correct methods for you. And then you'd simply drop the samples in at the mass spec facility, and we're on the ground floor of LIMS 1. Yeah, so that's the little workflow, and you've got our emails there as well for contact. And that brings my section to a close. And I think we can open it up for any further questions.
So, yeah, I'm going to discuss how we can help you with the data analysis side. And once Pierre and Rohan finish with the experiment on the instrument and the sample preparation wise. Pierre actually already went through this in a very brief way. So this is a little bit complicated way of presenting what Pierre and Rohan actually described. So what Pierre said was, yeah, you inject a sample into the HPLC system, and this is our mass spectrometer, and this does all the analysis, and at the end of the analysis, we get a result. And this result is actually, if you're familiar with genomics data, is slightly different to what we are getting from there, because at the end of the process, what you will get is a bunch of numbers. And we will not be able to give you, this instrument will not give you a list of amino acids. And then what we had to do is, in order to get the numbers, we had to do a bit more number crunching here, Fourier transform, and then plot them into and store them into a data file and output file. So different instruments we have will produce different types of data output files. And most of our instruments at the moment are thermo instruments. So these instruments will produce a file called the raw file. So that's their proprietary data format. And if you use a Bruker instrument, they'll have their own data format. Within the instrument, before we get the data, the instrument is doing a little bit of data processing, but it's not a lot. We'll do just a bit of centroiding and just to get the peaks out of the complex signal I showed you in the previous slide. So if we go back to what Rohan was discussing in the 4 scenarios, so you can actually use 2 main ways of analyzing this data. One is you can use de novo sequencing, and also you can use a search engine. So if you have a recombinant peptide or something, then if you want to know what this peptide is, then you can use de novo sequencing, and also if you know the organism, etc., then you might be able to use a search engine as well. But if you are talking about a very complex comparison, for example, comparing a wild type versus a mutant and the whole proteome, then we will not be able to use this technique because of the computational complexity of the problem. Then we will have to use the search engine. And it's the same for the quantification, but for post-translational modifications, it is actually in between, because mainly we will be able to use the search engine and get most of the information. I would say more than 95% of the time we will be able to get what we want. But there are situations where we, because of the limitations in the algorithms, we will not be able to get everything out. Then we had to use a combination of de novo and manual annotations as well as search engines. In the de novo peptide sequencing, what we do is it's a bit like sequencing your gene, like DNA sequencing. So instead of looking at the ATGC, we just look at what's the difference in the mass compared to the original mass of the peptide. So as the fragmentation goes on, these individual fragments get break off, and we can actually work out from the fragments what might be the full sequence of that peptide. This is actually very useful in situations like situations where you need to sequence something like a CDR3 region of an antibody. It's going to change based on the condition, and so you won't be able to have a definite sequence, but it's short enough, and we can use this technology for that. And also, if you are doing a synthetic peptide, like if you want to make sure you got the correct peptide, then we can use this method for this. And as I mentioned before, post-translational modifications can also benefit from this if you have troubles with the search engines. But in most of the cases, what we do is we use search engine called Proteomics search engines. So the vital part for the search engine is the sequence database. So without the sequence database, we will not be able to perform this search. And also, if your protein of interest is not sequenced, or if you don't know the exact sequence, then we will not be able to use this method. So what search engine does is it will take your sequence database. It's a bit like a genomics aligner. Instead of just doing a pure alignment, it will do processing on the database and create a theoretical set of spectra. And then once the theoretical spectra is created, then we can align this theoretical spectra with the actual spectra generated by the instrument. And then finally, we can do peptide and protein mapping. In our facility, we have a number of search engines available, and these search engines are different, have their different properties. And like Mascot is quite old, and it's actually a very good algorithm if you want to detect post-translation modifications, etc. And the Barnet is quite specific and also very good at detecting post-translational modifications. One important thing I want to mention is the data we are getting out of, or the results we are getting out of search engine is always has a probability value attached to it. So for example, if we say we detect your protein of interest at a high confidence or less than 0.01 false discovery rate, that doesn't mean you're 100% sure the protein this is the protein, which means, in other words, there's 1% chance there could be a wrong identification. This is purely due to the algorithmic issues with the mapping. And at the same time, if you drop the false discovery rate, your list of proteins will get longer, and at the same time, your chances of getting a wrongly identified protein will go up. So based on the situation and the application, we adjust this false discovery rate, but always we try to work at a very high confidence levels, which is at least 0.01 FDR. The other thing is these search engines, as I mentioned, these algorithms can have some impact on your results. Like for example, if you look at this is a publication from 2013 where they compared Mascot, Sequest, and MaxCount. So as you can see, there are common elements as well as there are subtle differences between the search engines. So it's what based on the problem we have, we will be picking the best algorithm for you, and we will do, if we need, we will combine different search engines as well. So Pierre also discussed briefly about the protein quantification, and so there are actually two types of quantifications we can do. And the most common type of quantification is the relative quantification, where we compare between samples. Like in this case, we'll be going across the rows, comparing sample 1 against sample 2 against 3, likewise. So this is a relative quantification. And also we can do absolute quantification, but absolute quantification is much more complicated than a relative quantification problem, because we'll have to work out various different parameters before we actually go ahead and do the experiment. And also, it's very difficult to do it for a large number of proteins. For example, if you are comparing two cancer cell types, you most likely won't be able to do this. But relative quantification can be very successfully applied in that situation. So I won't go into the details of the relative quantification, but if you're interested, this is a very good resource, and I think you find most of the technical information about the quantification process here. So to do the quantification, we have a number of softwares available, and MaxQuant is one of the most famous software to do this work, and it's the most cited software. And also we have proprietary software like Peaks and Proteome Discoverer, etc. Once the quantification is finished, then we can do downstream analysis, like for statistical analysis and also gene ontology analysis, etc. And also data visualization. We have the capacity of doing that using the proprietary software like Peaks, Scaffold, and Proteome Discoverer. But one of the limitations I can think of, I can point out, is these softwares are actually limited to what's available on the menu. So if the method that you're planning to use is not implemented in the software, or the method is not appropriate to your application, we won't be able to use any of this, then then we have to think about a different way of doing that. But to overcome such issues, nowadays programs like Proteome Discoverer are incorporating a programmable module, so we can insert some R code or Python codes and go around, but still it is very limited. In most of the cases, what I do is what I'm studying is from my head, so I have a lot of freedom to manipulate the data and get the insight very easily. And at the same time, I would like to introduce you to something that we have developed in our platform, which we call the CPP Analytics, or the Proteomics Bioinformatics Toolkit. This is a web-based tool that is freely available to everyone and can be used to do two different purposes. One is the data analysis, and the other one is the data visualization. So there's one app particularly dedicated for the analysis, and the others are dedicated for data visualization. So for the t-test, heat map, multidimensional scaling, box plot, correlation, they all require an input file. I mean, all these applications require an input file, but for these ones, specifically, we designed it around the most common and the freely available program called MaxQuant. So once we provide you this output file, you can simply provide it to this application, and you can use it on your own. But for the volcano plot, it doesn't have to be a MaxQuant output file, but any statistical output file will work. For example, if your t-test result has a fold change and just a p-value attached to it, you can use it. It doesn't have to be a proteomics experiment. It can be a genomics experiment. And upset plot is also the same. Actually, this is, I'm not sure how many of you are aware of this, but this is a different type of representation of sets. Like, let me see if I can draw something here. So you have sets like that. So once this Venn diagrams start going more than is going to occur, is going above more than 5 samples, this is going to get very complicated. So to go around that, we use this upset plot. Now, I would like to give you a little demonstration of how you can use this. So in the future, when you're okay to use this one, you can go ahead and use it. So I'll try to exit this presentation and come back to it in a second. Okay, so if you log into the CPP Analytics, this is what you get. This is our home page, and we have a number of applications available. So you just need to click on any of these icons. So it will take you to the application directly. So I'll give you a demonstration of how you can use a volcano plot application to draw a volcano plot. So it's accepting statistical result output, where you have the fold change and the p-values. So all you had to do is just browse there and then select the data. It will automatically draw it. So all you have to do is now, if this is for aesthetic purposes and for the publication purpose, if you are not happy with the default dot size, you can change it. And if you're not happy with the size of the axis labels, you can change that. And again, axis title, change like that. This is for you to format the figure. And this bar is to adjust the cutoff for the log fold change. So you can simply move it. Then you will see this bar is getting moved, and you can move the other way. Then you can see this bar is moved the other way. And if you want to change the p-value, again, you can move it like that. You can see this bar is moving up. And if you want to apply gene labels to those proteins or genes that are significant based on your parameters, you can simply click this. It will apply the gene names. And in the end, if you want to get the table, filter table, you can just go there, and then there you have the table. So this table will get filtered based on your on the parameters that you're putting there. And once you're happy with everything, you can download it using this button. This is for downloading the figure. So this will download the figure, and this will download the table as a CSV file. And the correlation plot is also the same. So pretty much it will require you to provide the output file from the maxcom. So it's called the protein group file. So you just go there and provide it. Okay. So yeah, then you can decide what clustering method you want to do and ordering methods, etc. For the multidimensional plot, again, it's the same. So I'll go through this very quickly. Just upload it. One additional thing you need to do here is you need to select number of experimental conditions and how many replicates do you have in each condition. And the heat map is also pretty much the same. So just go and upload the maxcom output file. Yeah, there you go. So you have the output file there. So you can select whatever the clustering method you want. Or if you want to zoom into a particular row, you can simply use that. So now you are zooming in from 1071 to 1174. So you can do it with the columns as well, like that. Again, if you want to download it, you can download it from there. And if your input file has gene names, if you click this one, it will let all the gene names to the rows. So I'll close this one and go back to my presentation. So in addition to what I just described and providing a statistical analysis, we can also provide you with advanced analysis. Like, for example, signaling pathway analysis. Signaling pathway analysis is mostly effective if you use phosphorylation, because up and down regulation of proteins doesn't really, there are a lot of evidence that up and down protein regulation is not directly representing an activation or inactivation of a signaling pathway, but phosphorylation and dephosphorylation is has a direct link with that. And also this is something we very recently set up in our lab. So this is the protein cross-linking. So in this case, we can use cross-linking methods to bind cross-link complex of proteins and identify what proteins are present in this complex. And we can go one step further with this. And once we identify what proteins are in this complex, we can also create three-dimensional complex models. This is simply by arranging the protein, by mapping how the proteins are arranged in the three-dimensional space. And this is quite useful if you are interested in doing drug design, or if you want to try and block one of the interactions. Or for example, if you identify a critical node here and you want to design a drug against, again, this is something quite useful. And we can also help you with that by helping you with high throughput in silico screening. So at the moment, there are large number of compound libraries, like going up to 200-300 millions of different compounds that can be purchased. So you can run through them and do some in silico screening there. Also, this is quite useful if you are doing studies to understand how your proteins are interacting with other proteins. And you can also do interactome analysis and also visualizations on that. Deep learning and machine learning is something we are also at the moment exploring. At the moment, we use deep learning models and machine learning models to do de novo sequencing. And also in most of our software, we are using them to do feature identification. And also you can use it to integrate data between proteomics and genomics, or transcriptomic analysis. Also, as a part of this whole thing, we can also provide you and help you with molecular dynamic simulations. Like, for example, this is a molecular dynamic simulation of transmembrane protein, which has quite a lot of implications on portal hypertension. And finally, imagine you came up with a brilliant idea of how to analyze a different analyze a data set, and now you want to share that method with others, or you would like to have you have a certain pipeline and you would like to share that pipeline with your colleagues. Then we can help you with the software development part and make that pipeline available to convert that pipeline into a software and make it available to others. So with that thought, I'm going to wrap up. If there's any questions, I'm happy to answer.
Thank you to our presenters today, Pierre, Rowan and Harinder. If anyone would like further information, please contact La Trobe University's Comprehensive Proteomics platform. Further information can also be found on the Proteomics platform website. Thank you for watching today's presentation.